Welcome to my poster on supervised machine learning reveals high efficacy of mobile elements to predict Salmonella outbreak linkage. So the motivation behind our study is that standard methods for predicting Salmonella outbreak clusters primarily involve the comparison of genetic variation in a stable segment of the pathogen genome, commonly known as the core genome. And as a result, non-conserved genetic elements are oftentimes neglected and don't really contribute any information to genomic comparisons. And so here we would like to demonstrate the analytical value of these non-conserved elements for outbreak cluster prediction. And so to do this, we trained regression models on known Salmonella outbreaks to characterize genetic features that are predictive of outbreak clusters. So the overall analysis workflow can be divided into two parts. The first part involves model training and feature selection. And this is to identify a set of features that are predictive of Salmonella outbreak clusters. We subsequently annotate these features to identify what functional elements in the Salmonella genome carry these predictive features. Here we use unitags as individual features to represent genetic variation in the genomic data. We then train elastic net regression models on these set of unitags, and we select the best performing models based on cross-validation. So over here on the top right, we're looking at the summary of the overall model performance, but specifically, we're looking at model performance in response to a hyperparameter called alpha. And I like to think of alpha in the context of regularized regression as the strength of selection. In other words, greater the alpha value, the fewer the number of features that are selected. And this is well represented in this violin plot here. So the numeric label at each alpha value represents the average number of features that are selected by the models at each alpha value. And what we can see here is that at an alpha value of 0, majority of the features in our data are selected and used for prediction on the test data. And what we see here is that as we retain majority of the features in the data, these models end up performing very poorly. And this is, in fact, a really good example of model overfitting, because we know that majority of the features in our data are not very predictive of outbreak clusters. And therefore, they're simply capturing random noises in the data, and this gets propagated to errors in the prediction. Now, as we start to select for a fewer number of features in the data, ideally predictive features in the data, as we increase the alpha value, we see a drastic improvement in the model performance. And what we see in this violin plot is that between alpha value of 0.1 and 1, these models perform equally well. And so rather than selecting a model from an arbitrary alpha value, we decided to aggregate information from all the models that we have generated in our study. And so in order to identify a set of predictive features from our data, we ranked each feature based on a score, and this score is calculated based on how frequent a given feature is selected by our models. And in the end, we find about 5,000 features to be consistently selected by the majority of our model. And we've then further characterized these 5,000 features through functional annotation. And so down below here, this table provides an overall summary of the functional elements that are mapped by these predicted unitags. And what we find is that about 80% of these predictive unitags map to protein coding sequences. And that's not too surprising, considering that over 80% of the Salmonella genome consists of protein coding sequences. But what is very interesting in this table is that about 30% of the predicted unitags map to mobile elements, such as genomic islands, bacteriophages, plasmids, and CRISPR repeats. And over here on the right here, when we look at the proteins that are encoded by the genes that are mapped by these predicted unitics, we find that a large proportion of these genes encode for hypothetical proteins. And if we look down this list, we find a lot of these proteins involved in bacteriophage assembly. Now, up to this point, we have only really answered the question of what functional elements are predictive of Salmonella outbreak clusters. And what we haven't really answered is how these functional elements differentiate the Salmonella outbreak clusters. And so we decided to then perform feature validation by analyzing 3 Salmonella Heidelberg outbreaks that are very closely related, as they could not be resolved by traditional molecular approaches such as PFGE and phage typing. And so what we would like to demonstrate here is that we can in fact differentiate these 3 closely related outbreaks solely based on variations in mobile elements alone. And so indeed we do end up finding unique molecular signatures in these mobile elements, including CRISPR arrays, phage elements, and plasmids. And specifically, we found that repeat copy number variations in the CRISPR arrays, single nucleotide variants in the phage elements, as well as unique set of plasmid carried by these genomes, can be used to differentiate these 3 different outbreaks. And so it is quite clear that there are signals in these mobile elements that can be leveraged to resolve Salmonella outbreaks. And the typing of these mobile elements can serve as an effective complement to existing methods that primarily focuses on the comparison of the Salmonella core genome.